Okay, question number one seems like it's pretty easy and repetitive. We've already covered the idea of Gibbs free energy in the energy chart. Remember, we have their x and y axes, and the x and y axes define what the graph is supposed to be informing you of. Your y-axis is your energy, and remember that your x-axis in this case is the progress of the reaction. Now, that being said, remember that you have a certain amount of energy that the molecule is starting at. Then you have an amount of activation energy that's required to start the reaction. And then there's the ultimate position at which the reaction, the products are going to be at. Remember, this side is the products, and the other side is the reactants. Now, it's this activation energy that's impacted by the enzyme. You have to remember that, because that's going to come up again and again throughout the rest of the year. It's this amount of activation energy that's lowered. The enzymes lower the activation energy by taking the reactants and creating conditions that allow the reaction to go forward with less required energy. So you have to remember that that is pretty much the explanation for how the enzyme impacts the chemical reaction and the energy chart for that chemical reaction. The answer for number two seems to be a pretty simple one if you think about it. If each and every reaction, if you have 100 reactions, 1, 2, 3, all the way through 100, if these 100 reactions are what are continually allowing you to be who you are, one reaction makes you feel good, one reaction makes you feel bad, another reaction allows you to move. Without the enzyme, that reaction won't go forward. So what your body does is that it produces enzymes in either greater or lesser concentration, allowing the reaction either to go forward or not to go forward. And it actually controls the process of the reaction, controls the reaction by controlling how much enzyme is being produced at any given time. So hopefully you got that idea from the lectures and the activities, and you were able to get those 10 extra credit points. Now here, in this figure, what you see is the enzyme binding the substrate at the activation site, and you see that the product being produced, in this case the splitting of one molecule into two separate molecules, is being done by the enzyme. The enzyme is not impacted, is not changed, does not actually chemically change with the reaction. So all the enzyme is doing is assisting the reaction to go forward by creating an environment that's chemically favorable for the reaction to happen, therefore reducing the amount of activation energy required for the reaction to occur. Okay, the next question then is asking something very simple, and that's something that you should definitely know, which is what are the functional groups that are involved in these reactions that you see above. And the functional groups are those that I asked you to memorize several times. This here is your carboxyl group. That's COOH or C double bond O, single bond O, H. Now I saw some people put COH, I let that go just because you came closer than a lot of people did. But for your next quiz, which will be on Monday, please try to make sure that you study these functional groups and know what they are. The other functional group that helps define an amino acid, remember, it's an amino acid. This is a carboxylic acid group, so that gives it the acid name of the amino acid. The amino group is the NH2. NH2 is an amino group, and that's where amino acids get their first name. So the amino acid comes from the amino group, and the acid part of the amino acid comes from the carboxylic acid group. So when you're dealing with water being produced, that means that water is leaving the system. If water is leaving the system, then you have to think of the word dehydration. Remember, dehydration means the removal of water. Notice that you have here in the little red selected area one OH, one H, which makes one H2O. The H2O has left the reaction, so this is a dehydration. And of course it's making a polymer, so it's synthesis. So it's dehydration synthesis is the answer for number 5. Number 6 was one of those things that was really easy if you studied. It's what are the bonds that hold the two things together. This was a peptide bond. That's again peptide bond. It was going over and over again in both the notes and the video. And a peptide bond holds the two amino acids together. So an amino acid is held together to another amino acid by a peptide bond. I gave half credit for the word covalent because a peptide bond is a type of covalent bond, and it's just a covalent bond that holds together two amino acids. And specifically, if you really wanted to look at it, you can look at it. It's the bond that's formed once the OH and the H leave the two amino acids. Why are proteins often called polypeptides? Because proteins are a series of amino acids linked together by these peptide bonds, and it could be hundreds, if not thousands, of amino acids long. So because of that, there's, of course, polypeptides, or more than one peptide. Polypeptide. So that's why it's called a polypeptide. A protein is a polypeptide because it's made of amino acids. It's a polymer of amino acids, and it's the peptide bond that holds them all together. Number 8, even though it's pretty long, is really, really, really simple. There's 20 amino acids, and if you look at your notes, you'll see that all 20 were listed, and there was a group that was hydrophobic, and there was a group that was hydrophilic, and each type of amino acid, there were a bunch of them, but the bottom line was that there were 20 amino acids. Each one could take the place of an R group in the basic structure of an amino acid. Okay, number 9 is pretty straightforward. It says, what is the sequence of DNA above? If you're going to read from 5 to 3, remember we said that that's the direction that DNA and RNA is read from 5 to 3. So if you're going to read it from 5 to 3, it would be T, C, G, A. So that's the sequence. T, C, G, A. What's really cool is if you read the red strand, the red side of the strand instead, again, reading it from 5 to 3, then you get T, C, G, A. It's the same sequence because they're anti-parallel, or they're parallel, but they're opposite codes, therefore anti-parallel. You have the same sequence on both sides. Now, if you read 5 to 3 for the RNA, then you would, which is the answer to number 10, then you would see U, C, G, C. Now, it's important to note here because in the next two questions, you were asked to look at what's the difference between RNA and DNA. Please notice that in DNA, there is no oxygen here. The oxygen is missing in DNA, hence deoxy, where in RNA, the oxygen is there. You see it there in the same position over and over again. So, okay, you saw the RNA sequence from the slide, the previous slide just a moment ago. So we know what the RNA sequence was. If not, rewind it and look at it again. What are the 3 parts of a nucleotide? Again, right out of the notes, right out of the video, it's the nitrogenous base, it's the phosphate, and it's the ribose sugar. Now it could be either ribose or it could be deoxyribose, but really it just is a pentose sugar, right? It's a sugar with 5 sides. What does the pentagon shape above represent? It was that pentose, that ribose sugar, that pentagon shape that gives you that's part or half of the sugar phosphate backbone of DNA or RNA. What are the two differences between RNA and DNA? We already discussed it earlier, and that is that oxygen in that 2' position, and the fact that you have uracil in RNA, or in DNA you have thymine. All right, hope you got that.